Hello, everyone. Thank you for joining this professional training session on the ICH Q3D Guidelines for Elemental Impurities. Now, let's go through today's presentation, which has been carefully developed to provide a comprehensive and practical understanding of the Q3D principles, their applications, and regulatory expectations. Whether you're from a regulatory agency, pharmaceutical company, or quality control lab, this session will enhance your grasp of the guideline and how to implement it effectively in your daily work. The ICH Q3D Guideline is a harmonized standard that establishes acceptable limits for elemental impurities in drug products. It provides a risk-based approach that helps ensure patient safety while supporting manufacturing efficiency. Elemental impurities may come from various sources: raw materials, processing equipment, water, or even packaging components. The Q3D guideline applies to new drug products across all ICH regions and considers the route of administration, oral, parenteral, and inhalation, as a critical factor in safety evaluation. In simple terms, Q3D helps us manage the risk of toxic metals in our medicines. To support implementation, the Q3D guideline is accompanied by structured training modules. Module 0, which we are covering now, provides an overview of the entire framework. Modules 1 through 7 delve into technical aspects such as safety assessment, calculation methods, and control strategies. Module 8 offers real-world case studies, and Module 9 responds to frequently asked questions, helping to clarify common misunderstandings. This modular setup allows professionals from diverse roles to focus on the sections most relevant to them. At the core of Q3D is the concept of the permitted daily exposure, or PDE. These values are based on toxicological data and represent the maximum acceptable intake of a particular element without appreciable health risk. The guideline includes PDEs for 24 elements, with separate values for oral, parenteral, and inhalation routes. If a drug product may contain levels above the PDE, we must provide scientific justification, often using animal toxicology data, clinical safety data, or both. The guideline encourages a balance, protecting patients without unnecessarily burdening development. There are two common approaches to risk assessment: the drug product-based approach, which assesses the final product as a whole, and the component-based approach, which evaluates each ingredient, equipment, and process step. We examine potential EI contributions from drug substances, excipients, manufacturing equipment, water, and container closure systems. By using supplier data, historical results, and validated methods, we determine whether additional controls are necessary. The goal is to proactively identify and manage risk sources before they impact product quality. Once we've assessed the risk, we develop a control strategy. Preferably, controls should be applied upstream, through quality agreements with vendors or material specifications. Testing the final product is a last resort. If a component consistently meets low EI levels, we can avoid routine testing altogether. This lifecycle-based approach also means we must periodically reassess our strategy, especially if there are changes in suppliers, equipment, or regulatory expectations. Modules 3, 4, and 7 address specialized topics. For instance, Module 3 covers how to assess elements not listed in Q3D using toxicology data to derive internal PDEs. Module 4 focuses on large volume parenterals, which are more critical due to higher exposure risk. The guideline permits a conservative 2-liter assumption for dose calculations. Module 7 guides us on converting PDEs into concentrations using options 1 through 3. This enables alignment between risk assessments and analytical specifications. Nothing explains theory better than real-life examples. In Case Study 1, a parenteral protein drug was assessed using batch data and raw material screening. Most EI risks were mitigated through supplier controls. Case Study 2 involved a synthetic small molecule called Great Drug. This injectable product was assessed for RH and PD from catalysts and PB from glass vials. With effective purging and qualified packaging, risks were minimal. These examples demonstrate that Q3D is not one-size-fits-all. It's about context and risk-based flexibility. Q3D often raises recurring questions. For example, why aren't herbal medicines included? Because of their complex, variable nature, they require a separate regulatory approach. Aluminum is another question mark. It's excluded since its toxicity profile and natural occurrence differ from other elements in the list. As for the 24 elements, they were selected based on global pharmacopeal guidance and observed risk in pharmaceuticals. These FAQs help refine our understanding and application of the guideline. To wrap up, ICH Q3D is a robust, science-driven guideline that balances safety with practicality. Its training materials, risk frameworks, and examples support effective, harmonized implementation. Whether you're in R&D, manufacturing, QA, or regulatory affairs, understanding Q3D empowers you to develop safer, compliant, and high-quality medicines. Get the ICH reference documents in description below for your reference. Thank you for your attention. I'm now open to any questions or discussions you'd like to have.